Good morning and welcome, everyone. I'm excited to introduce our two speakers today, Dr. Anne Burchell and Dr. Curtis Cooper. Dr. Cooper is a professor of medicine at University of Ottawa's Department of Medicine and is an infectious disease consultant. He's also a senior scientist with the Ottawa Health Research Institute and director of the Ottawa Hospital and Regional Viral Hepatitis Program. Dr. Burchell is a scientist at the MAP Center for Urban Health Solutions, St. Michael's Hospital, Unity Health Toronto, and Associate Professor, Department of Family and Community Medicine at University of Toronto. Her research program focuses on the epidemiology of HIV infection, HPV, syphilis, and other STIs. She's the principal investigator of the CHEST study, which is evaluating the burden of COVID-19 among people living with HIV in Ontario. Also joining me today is my colleague Eric Peters, who will monitor the chat for questions. Please write any questions you have in the chat. We will take the questions after the presentation. And a big welcome to Dr. Curtis and Dr. Anne.
Great. Well, good morning, everybody. It really is a pleasure to join you today and chat a little bit about COVID immunity and some of the research activity that's ongoing in Canada. Special thanks to Devin for the invitation. It really is appreciated. I will pull up my slides here. I hope everybody can see these.
We can't see them yet. Can you try resharing?
Sure.
I can also share your slides, Curtis, if you'd prefer, and you can just tell me when to move ahead.
Yeah, I'm not sure what the trouble is here. Okay, maybe in the interest of time, why don't I ask you to take over the slides? There we go. Good.
You can just tell me when to advance.
Sure. So, as I said, we're going to talk about COVID immunity and people living with HIV. Next slide. These are my disclosures. Next slide. We're going to review how people living with HIV develop immunity to natural COVID infection and to COVID vaccine. We'll discuss COVID vaccines currently in use in Canada. And then we'll review several key Canadian research studies that are focused on the intersection between COVID and HIV. Next slide. So this is a little diagram of the life cycle of COVID. The virus comes along, interacts with the cell membrane using, in part, the ACE2 receptor. It gets inside of the human cell. The virus is broken down into bits and pieces, which then are delivered to various parts of our immune system. And while all this is happening, new virus is made within the cells, and then a virus is expelled. New slide. The key part of the virus, and I suspect you've all heard about this protein, it's the S protein or spike protein. It sticks off of the side of the virus, and this is a key target that our own immune system will recognize and develop an immune response to. And a lot of the vaccines are focused on using this spike protein in order to develop immunity to protect us from COVID infection. Next slide. So how is immunity developed? Well, as I suggested, the virus comes along, it binds onto one of our immune cells, the virus is taken in within that cell, it's broken up into bits and pieces, which are then expressed on that immune cell. There's two key families of immune cells in our body. There's T cells, some of which are responsible for going along and ingesting pathogens that shouldn't be there and breaking them up and destroying them. So these are the CD8 T cell or killer cells. And then there's other T cells that ingest components of the bacteria or virus or fungus that the person is infected with, and they'll start to produce chemical messengers called cytokines, and these cytokines will tell the rest of our immune system how to react. It'll tell the immune system to make more of this particular kind of T cell, or it'll tell the immune system to make more B cells to produce antibodies. So the other family of immune cells are the B cells, and these produce antibodies which help to protect us from infection. These antibodies are produced, and then when an infection that shouldn't be there in our body arrives, these antibodies bind to it and then help to clear that infection. Next slide. As I suggested with the T cell immunity, they produce immune cells and they produce a whole bunch of different cytokines. Remember, those are the immune messengers or chemical messengers produced in our body. So examples of these include interferon. So when you're feeling achy and sore from an influenza infection, part of that is because your body is producing interferon to tell your immune system to fight off the influenza. But one of the unfortunate consequences is that you feel feverish and achy and sore because of that interferon. And we also used to use interferon to treat hepatitis B and hepatitis C. So some of these cytokines we put to work for medical practice as well. Next slide. Now, if and when you're reviewing studies looking at T cell production from a vaccine, there's many different ways that you can assess how the T cells are working. You can look at their absolute numbers. You can look at the T cell ratios. There's lots of different T cells. You can look at how many cytokines or how much cytokines these T cells produce. So in short, there's lots of different ways that we can assess T cell function. Next slide. And then remember, the other family of immune cells are the B cells that produce antibodies. There's lots and lots of different kinds of antibodies that are produced depending on the infection and depending on the scenario. The ones that we're most interested in are the IgMs, which are produced early on during an infection, and the IgGs, which are produced shortly thereafter. And it's those IgG antibodies that help to produce a long-term protection against infection. Next slide. When we're looking at when we're looking at COVID vaccine studies or any other vaccine studies for that matter, you'll often run across graphs that look like this, where they'll show an initial spike in the antibodies, usually mostly IgM, and then a second spike, which is mostly IgG. And again, it's these IgG levels that will hopefully persist for a long time and produce long-standing immunity. Okay. Another key point is we're really focused on antibodies when we're trying to decide whether we're immune to something or not. And the reason we're focused on antibodies is they're a lot easier to evaluate and measure than cell-mediated or T-cell mediated immunity. But just because your antibody levels may be low, or even if they're non-existent, doesn't necessarily mean that you don't have some level of retained immunity. So this is really relevant to people who have had natural infection with COVID or who have received vaccines for COVID and they notice that their antibodies start to decline to low levels. This does not necessarily mean that you no longer have any immunity. Next slide. And again, there are lots of different ways to measure antibody immunity. We can look at absolute levels of antibodies before, during, and after immunization. Next slide. We can also look at assays to see how well these antibodies will block and destroy infecting viruses or bacteria or whatever else we're looking at. So again, lots of different ways that we can measure antibody function. Next slide. So that was a primer in how our immune system works. Again, two major families of immune response: the B cells, which produce antibodies, and those antibodies are really important in providing long-term immunity. And then there's T cells, some of which are killer T cells, which ingest invading infections and get rid of them, and other T cells that produce cytokines, which are chemical messengers to tell our body's immune system how to fight off infection. Just briefly, lots of activity with developing vaccines for COVID these days, and there's lots of different types of COVID vaccines. We're probably most familiar with the RNA vaccines. RNA are little biological codes. They get taken up within our immune cells. This code is read. It produces proteins, so the spike protein, which our body then recognizes as foreign and produces B cells and T cells to protect us against it. And as you know, we currently have two vaccines that we're using in Canada that use this kind of strategy. There's also the AstraZeneca vaccine, which uses a different viral vector, so an adenovirus vector. That adenovirus gets into our body through the vaccine. It gets into our immune cells, and then there's a little piece of the COVID virus that's in that adenovirus, and again, it's produced within the immune cell, and then that protein is used to generate immunity. Next slide. So again, three different key viruses that we're using in Canada right now. The adenovirus AstraZeneca one, we're not using that much of that anymore. And the two workhorses are the Moderna RNA vaccine and the Pfizer RNA vaccine. Next slide. And I'm not going to talk much more about the specifics of those vaccines. We can cover that during the discussion if people have specific questions about that. Now, I think we're all keenly aware of the fact that there's not a lot of literature out there about how these vaccines work in people living with HIV and issues related to safety. In fact, the best data we have is with the AstraZeneca adenovirus vaccine, where they did include some people living with HIV in some of their South African studies. So this was an early phase study conducted almost a year ago, where they looked at adults between 18 and 65 years old. And in group 3, they did include people living with HIV. Next slide. Individuals received the vaccine at baseline and at 28 days, and then they assessed for a year afterwards safety measures, and they did serial blood draws to look at the immunity response. And again, they looked at antibody levels, antibody neutralizing function, and they looked at various measures of T cell function, so cytokine production and how well they work at killing foreign viruses. Next slide. So ultimately, they dosed about 102 individuals living with HIV. Next slide. The individuals living with HIV had a fairly high burden of comorbidities. So 8% had high blood pressure, 10% had underlying lung conditions, 3% had evidence of diabetes, and 22% had a higher BMI. And most of these participants in this particular study were of Black ethnicity. They were on antiretroviral therapy with suppressed viral loads. So all that's important to understand the population that they studied. Next slide. And they found that the vaccine was just as safe and just as well tolerated in people living with HIV. Next slide. And they found that the antibodies generated, the levels were very similar to people who were not living with HIV. Next slide. And similar comment for people as far as their people living with HIV, as far as their next slide. Oh, sorry, I guess I cut that slide out, but the T cell responses look to be about the same as people not living with HIV. So with this particular vaccine, it was found to be safe and generated an immune response that was similar to people not infected with HIV. So that's great, except it leaves out an awful lot of people. So not focused on people living with HIV who are aging. Remember, the cutoff age was 65. Didn't look at immune response in people not currently on antiretroviral therapy or with viral loads that weren't fully suppressed. Also, this is the data related to one vaccine that we're not really using in Canada anymore. So there continues to be a fairly large void of evidence to help guide our use of COVID vaccines in people living with HIV, our level of comfort as far as safety, and then bigger questions that this study didn't even contemplate are questions related to, do we need booster doses beyond the first two? So next slide. So I and Burchell and many others are pursuing studies trying to address some of these issues. I'm first going to talk about the Stop the Spread cohort that I and Marc-Andre Langlois, who's a basic scientist with the U of Ottawa, have developed. And then there's a number, many other investigators who've contributed to this. Next slide. So the next slide. So Stop the Spread is a 1,000-person cohort of individuals who have either had COVID or were at risk for being exposed to COVID. And we did baseline questionnaires and drew baseline blood work in these individuals. And then they've been coming back every month for 10 months for repeat blood draws. And we've been completing questionnaires at 3 and 10 months. Okay, many of these people have now got to the 10-month point, and some of them are being enrolled in a longer-term 2-year follow-up where we collect blood every 2 months. And we're collecting this blood to look at their immune response over time to natural infection or their immune response to receiving vaccination. Next slide. Our cohort is characterized by being about 70% female, about 85 to 90% white, but 10 to 15% of other racial groups. There's a fairly heavy representation of other comorbidities. So we have 24 individuals who have cancer. We have 41 participants with diabetes, 30 living with HIV, 74 with other immune deficiencies, 141 with obesity, and then other subgroups with other comorbidities. So ultimately, we have 1,000 person group of individuals that have individuals who are young and healthy. We have people who are older and healthy, and we have people with multiple different comorbidities. Next slide. And we're using this data set to address many different research questions, which include what is the antibody response in people living with immunodeficiencies compared to immune competent individuals. We're looking at that ACE2 receptor that the COVID vaccine uses to get into the cell to see if we can understand how the spike protein binds onto that. We're using this cohort to assess the predictors and outcomes of people with long COVID, so people who continue to suffer from side effects of the infection long after the time of the infection. And we have probably about 150 or 200 people with identified long COVID symptoms. And the list goes on. Now, importantly, we're using this cohort as a control population to compare to other populations, including people living with HIV. So next slide. Which brings us to the COVAX HIV study, which is a Canadian HIV Trials Network trial, specifically looking at a cohort of people living with HIV who have received or are in the midst of receiving vaccine for COVID protection. Next slide. Now, Cecilia Kostanek is the big lead on this study. I'm here as a senior lead, and then Aslam Anis obtained the funding from the Canadian Immunization Task or Immunity Task Force. And this is a list of many of the investigators who are contributing to this study. Next slide. So, as I've already suggested, there is evidence that people living with HIV have less robust immune response to vaccines, especially if they're not on antiretroviral therapy and if they have low CD4 cell counts. And as I've suggested, there really is a paucity of data looking at the efficacy of COVID vaccines in people living with HIV. Next slide. So our primary objective is to study the antibody response at intervals before, during, and after participants receive COVID vaccine. Next slide. So there's key subpopulations that we're looking at in this study. So people living with HIV who are older, so over 55. We're looking at, and recall with the scant literature that is out there, nobody really looked at older people living with HIV. And we're also looking at populations with lower CD4 cell counts and unsuppressed HIV viral loads. We're looking at people living with HIV who have lots of comorbidities. And then we're also, of course, looking at people with so-called stable HIV, so fully suppressed HIV, viral load, and higher CD4 cell counts. Next slide. It's a multi-center study. We're almost at our target of 400 people living with HIV. We have recruited in Vancouver, Toronto, Montreal, and in Ottawa. And we're collecting this fantastic collection of data and blood specimens that we'll use to assess immune response and safety to the COVID vaccine. And then we're going to use my 1,000-person Stop the Spread Ottawa cohort as a control group so that we can look at the immune response in healthy people not living with HIV and compare it to the individuals participating in this CTN study. Next slide. We're collecting specimens before the vaccine is received, 4 weeks after the first dose, 3 months after the second dose, 6 months after the second dose, and then 12 months after the second dose. That longer time point will allow us to assess the rate of decline in antibody levels and compare that using the Stop the Spread Ottawa cohort to see if there's similarities and differences between people living with HIV and people not living with HIV. Next slide. So as I said, we're getting really close to our 400-person target. And in our cohort, we have a good representation of men and women and people over the age of 55. We have patients with low CD4 cell count, or sorry, we have participants with low CD4 levels. We have about a third who have lots of comorbidities. And then we have at least a quarter who we would include in our stable and healthy HIV group. So given the numbers, we're going to be able to hopefully answer a lot of questions that pertain to a broad spectrum of people living with HIV and not just sort of that narrow group of perfect study candidates that we often get with our clinical trials. Next slide. So we're 75% complete with our enrollment. We already have a paper under consideration describing our protocol. We're working on trying to get additional funding so we can extend this study beyond a year. And we hope to start having data available for the community and for the academic community as well, starting in the spring. And I do want to very much emphasize that right from the start, when we hatched this study idea, we've been working hand in hand with community advisors. So I hope that you'll find that this study reflects the input and the needs of the community as well. Next slide. So that, in a nutshell, is a primer in immunity overall, a discussion of the vaccines that we currently are using in Canada, and some of the key HIV-focused COVID vaccine studies that are occurring right now. So with that, I will stop, and I look forward to hearing Anne Burchell's talk.
Thank you, Curtis. That was a great review of immunity. I enjoyed that. And thank you, Devin, for inviting us to speak with the group today. And I'm looking forward to any questions that may arise towards the end, either in the Q&A or offline. So let me share my slides. Are they showing up okay?
Yes.
Okay, great. So I'm going to be talking I'm going to step back a little bit and talk about some of the, I will come with end with some remarks about COVID vaccine effectiveness, but I also want to pull back a little bit and talk more about some of the questions that might arise when we're thinking about this intersection of COVID-19 and HIV. There's a lot of things that you might be asking yourselves, communities asking, the academic community. So here are a few listed here. So one of the first things that people wanted to know at the beginning, before the vaccines were even developed, was compared to people who are HIV negative, or people living with HIV at higher risk for infection with SARS-CoV-2, or once infected at higher risk for severe disease or hospitalization. Are they a high risk population to be prioritized for vaccine delivery? Also, did the pandemic itself and all the restrictions imposed by the pandemic impact HIV care engagement? We could be thinking about all these deleterious effects that might be happening on making sure people are getting access to care, are well engaged in care, and have suppressed viral load and better quality of life. Once the vaccines come out, it's great to have an effective vaccine, but you want to make sure that people get access to it. So are people living with HIV able to get vaccinated in a timely way? And then finally, are these vaccines going to work as well for people living with HIV as they do for people who are HIV negative? And there's a number of different ways we could be looking at how vaccines work. So I'm going to highlight a little bit about answers to those questions, and then end with describing an ongoing study that Curtis has started to talk about. So in terms of risk for SARS-CoV-2 and COVID among people living with HIV, there have been a number of studies that have looked at this. To date, there really isn't strong and consistent evidence that people living with HIV have a different rate of infection with SARS-CoV-2 than HIV negative people do. There's a number of different studies that have been done. Some show a higher rate of infection, others a lower or an equivalent rate. So there isn't really a strong signal here of a different risk of getting it. But once infected with SARS-CoV-2, there is a growing evidence base that people living with HIV may be at higher risk for the severe COVID outcomes. And one of the ongoing questions that we're trying to figure out as epidemiologists is how much of that increased risk for severe COVID outcomes is due to HIV in and of itself, and how much of it is due to, say, the other comorbidities that people living with HIV tend to have, or the social determinants of health that we know are also affecting who gets more severe COVID. So I want to show you results from two very large studies. To date, there really haven't been similar studies looking at COVID outcomes among people living with HIV in Canada, but I wanted to show you two studies, one from the United Kingdom and one from the US, that have been very large population-based studies. So they aren't affected by potential biases in who volunteers to be in a study or not. So these data here are coming from the United Kingdom, where they looked, this is a very large study of over 17 million people who are attending primary care in the UK. And they were able to look at COVID mortality among people living with HIV and compare it to people who are not living with HIV. You can see on the right-hand side of the graph, this is cumulative mortality among, in blue, people living with HIV, compared to in red, people not living with HIV. So you can see already those crude differences in mortality rates, quite dramatic. And when the authors here were very careful in making adjustments for a number of different things that might confound that comparison between people with HIV and people without. And they found that depending on what they adjusted for, the higher risk for COVID mortality tended to persist. It was either a doubling of risk or a tripling of risk for people living with HIV. And they found that that higher risk was particularly heightened among people of Black race or ethnicity, where it was over 4 times, mortality was over 4 times more likely among Black folks who are living with HIV. Now, if we go to this other study now from the United States, this is a very large study, over 1.4 million people who are involved in this. Largest study to date, really looking at this from the US. And in there, they were able to look at around 13,000 people living with HIV and compare a number of different COVID outcomes. And they did a lot of adjustment. So they're being very careful about making sure adjusting for differences in age, race, ethnicity, smoking, comorbidities. And they find this persisting higher risk of death due to COVID, about a 1.4 times higher rate, a higher risk of hospitalization with COVID, and a higher risk for severe COVID, but an actual lower risk for mild or moderate COVID. And then they did some in-depth analysis among people who were living with HIV specifically. Those are the bottom two rows here. And they found that really a lot of this heightened risk for severe COVID outcomes is among people with lower CD4 cell counts, most notably for less than 200. Not really a difference according to whether or not someone's viral load was suppressed. And in this study as well now in the US, they found that these heightened risks for COVID outcomes, again, tended to be particularly pronounced among older people, males, and racialized communities, specifically Black or African American adults and Hispanic or Latino adults. So I'm going to move now to another way that COVID might be intersecting with HIV, and that's starting to think about how the pandemic has influenced HIV care engagement in the cascade. So these are data that are demonstrating just what's the HIV antibody testing rate been like. And these, I'm showing specifically data from 9 different European countries that were combined. And what they're showing is in the same calendar period, comparing 2020 in red to 2019 pre-pandemic in blue, you can see a fairly dramatic decline in the red, solid red, in the number of HIV tests that were done. And when you go in and look at test positivity in the dashed line, you can see that test positivity in general was going up. So fewer tests were done in 2020 compared to 2019, particularly in the period of April and May, very soon into the pandemic. And the people who did come forward for testing tended to be at higher risk. And this kind of pattern is borne out in other places that have looked into it. Then we can think about how the pandemic restrictions might have impacted care attendance. And so we know here in Ontario and in other places, there's been a real shift to virtual care. So these are data from a large HIV cohort in the States, population-based among people who are veterans, so the Veterans Affairs cohort. And then they found, comparing 2019 to 2020, in all, there was actually an increase in the number of clinic visits that people attended for HIV care, but a lot more of the visits switched from virtual to, from in-person in the orange, to virtual in blue. So they went from about 1 in 4 being virtual in 2019 to about 2 in 3. And they found in this cohort that access to ART was not disrupted. That's not true everywhere, particularly in low and middle income countries. But in this cohort, it was not affected, and really led the authors to pose the question, could this long-term shift of virtual care be actually beneficial for some community members? The OHTN cohort study, which is an HIV clinical cohort in our province in Ontario, has also looked at this among people who've continued to attend their interview during the pandemic. And they do find that it's a minority of people who are reporting deferral of care, but it is not it's not a zero, it's a non-trivial number. So 18% of people said in Ontario that they had deferred getting their HIV blood work. And there are a number of other different aspects of HIV care here that were deferred. So it's a very important thing to keep watching. I talked to you about access to vaccines. So we'd want to know for people living with HIV, whether they're at equivalent risk for COVID, or we've certainly seen data suggesting higher risk. Some of them will, particularly those with lower CD4 cell count, for severe COVID if infected. We want to make sure that they're not experiencing any access problems in getting vaccinated. So here, I'm really pleased to share these data hot off the presses, courtesy of Sagaya Bekele and Abby Kroc at the OHTN. And they've shared with us here what has been the uptake of COVID vaccine among people living with HIV in Ontario. And what you're seeing here is, depending on the month that different people were interviewed, so these are different people in each bar, how many reported not having had a vaccine in gray, to having 1 dose in light blue, 2 doses in that middle tone blue, and now 3 doses in the dark blue. And you can see there was a fairly dramatic uptake that happened, so that by the time you got to August onwards, around 90% or so had had 1 or more vaccine doses, and most of them had actually had 2 or more doses. And this is quite comparable to the Ontario data. So when you look at all Ontarians aged 12 or more, or even if we just focus on Toronto, where a large proportion of people in this cohort live, very comparable. And already starting to see uptake of 3 doses. So Curtis spoke a lot about how we could look at antibodies and use that or T cells, and look at that as a measure of how well or not well someone may be responding to having been vaccinated. And that's important work to do. One challenge, though, with that is to date, we don't know what the correlative protection against infection with SARS-CoV-2 is. So we don't know, there's not a magic number that we can say, if you have this much antibody or these kinds of antibody present, you're good, you're protected. And differences in the immune responses or antibody levels, they may affect how well a vaccine will protect you, or they may not. So one of the things we also want to look at, in addition to antibody response, is vaccine effectiveness. So that is different from vaccine efficacy that we measure in a tightly controlled trial. This is going out into the real world once vaccines start being used and say, is this vaccine able to prevent outcomes in the real world? And a number of different places have now started to report estimates of vaccine effectiveness, including in our province in Ontario. I'm sharing with you results here from a paper published a few months ago from Jeff Kwong's group at the ICES, formerly known as the Institute for Clinical Evaluative Sciences. I'm going to talk a little bit about these findings, but the point that I'd like to make is, to my knowledge to date, and believe me, I keep looking regularly, there's not been an estimate of vaccine effectiveness yet, specifically for people with HIV. So in these data that are being shown, this was looking at how well vaccines were working against COVID outcomes between December 2020, when the vaccines started being rolled out in Ontario, to April. And they basically were finding that when you start looking at time from your first dose and then looking at after the second, so that's in the purple, to time after the second dose in yellow, you see pretty good vaccine effectiveness, particularly after the second dose against symptomatic SARS-CoV-2 and against hospitalization and death. So quite high, definitely above 90%. And those authors have subsequently continued to look at this up until August of this year. And they have a preprint demonstrating that this consistent high vaccine effectiveness, actually best for the mRNA vaccines, but also good for people who've had 2 doses protection across the vaccine products and across variants. But again, it's up until August. We need to keep monitoring it. And what we really like to see is a vaccine effectiveness estimate for people living with HIV. So these are a lot of questions we have. There's some data from other places, not a lot from Ontario, and we want to actually be monitoring this in our home province. So the CHESS study, I'm working with Curtis on this, along with Abby Kroc and Marc-Andre Langlois, and a great number of other investigators and community advisory board chaired by Devin. And we're doing what's called the COVID-19 HIV evaluation of serology and health services study. And our overall goal is looking at the burden of COVID among people living with HIV in Ontario. We're doing 2 different things, a dry blood spot seroprevalence study, and a lot of work we're going to do looking at health services using the administrative health data that are housed in ICES. So I'm going to tell you a little bit about what we're trying to do with this study. Because we're going to go into ICES, this is where everybody with an OHIP card in Ontario has information stored there that allows us to look at health outcomes, health services. So there's almost 12 million adults with data in ICES that we can be looking at and comparing to people living with HIV. Ontarians living with HIV, and we estimate about 23,000 And then we can also, working together with Abby Kroc and the OHTN, look at people living with HIV or participating in the OHTN cohort study, because those data are available at ICS. And then we're going to have about 1,000 people who are members of the OHTN cohort study or participants who we hope 1,000 will agree to give us dry blood spots that we can look at antibodies to SARS-CoV-2. And there's 4 main aims that we want to look at. We want to know what the seroprevalence of SARS-CoV-2 antibody is among people living with HIV in Ontario, which will allow us to get an estimate of how many have had natural infection and how many have present antibodies that have been vaccine-induced. We want to look at the burden of COVID-19 related analysis. We'd like to do the same kind of analyses that I showed you from the UK and from the US to see our rates of hospitalization or COVID death or symptomatic SARS-CoV-2. Are they higher for people living with HIV in Ontario as well? And then the last thing actually, the second last thing we want to do is the impact of pandemic on HIV care engagement. How has that shift of virtual care worked? And then end on a lot of work looking at vaccine effectiveness against lab-confirmed infection with SARS-CoV-2. And this last aim here, we're working together with the CTN-funded study, COVAX HIV. So we've paired up with them. So I'm going to talk a little bit about how that pairing up is going to work and how we really hope to have quite good data going into 2022 to answer these questions about COVID vaccine effectiveness for people living with HIV. We want to look at against lab-confirmed infection, hospitalization due to COVID, and we hope that we're going to be able to get deep into these data and look at, are there differences by sex, by age, socioeconomic status, comorbidities, and HIV immune status. So in addition to the work in Ontario, we've teamed up with British Columbia, and they have data at the BC Centre for Disease Control. Sina Samji is leading this work. So we're going to be able to do population-based studies of 9,000 adults living with HIV, we think, in BC, and 23,000 adults living with HIV in Ontario. Very good data should be available to us to look at COVID vaccines uptake and results from PCR tests for SARS-CoV-2, hospitalization, mortality, and then we will be able to adjust for a number of different things. In particular, in Ontario, we'll have quite a bit of data available to us to look at things like viral load, CD4 cell count, and in the subset of people participating in the OHTN cohort study, very individual level record information about social determinants of health. So what we hope we're going to get out of this particular analysis is really good assessment of what has been the uptake of COVID vaccination, vaccine products, timing, number of doses. I showed you that data from the OHTN cohort study. Those are volunteer members of the cohort who attended an interview during the pandemic. They may or may not be different from people who aren't in a cohort. So we'll be able to look at this population wide. And then we'll be able to look at very fine estimates. We hope the data will allow us to do this, to estimate the ranges of vaccine effectiveness of people with HIV, which will be very important for knowing what to do in the years to come around subsequent doses, for example. So we hope to have interim results available for sharing in winter and spring of 2021. And then we're going to repeat this analysis a year later, where we think we'll have a lot more data around the third doses, where we would share in early 2023. So what I've kind of tell you what I told you. Compared to HIV negative people to date, there doesn't seem to be a strong or dramatic difference in the rate of SARS-CoV-2 infection in and of itself, but the evidence does suggest that people living with HIV are at higher risk for severe outcomes if once infected, particularly if their CD4 cell counts are low or comorbidities are present. The pandemic restrictions and this expansion to virtual care might have improved the HIV care engagement. It may have worsened it. It's probably a little bit of column A and a little bit of column B, depending on differences in that we know people living with HIV aren't all the same. So we really need to look at this in Ontario going forward. To date, not seeing any evidence that there's been an equitable uptake of vaccine among people living with HIV, but we need to keep monitoring this. And then ongoing studies like CHESS and COVAX HIV are going to be useful tools to investigate whether COVID vaccines work as effectively for people living with HIV as they do for HIV negative people. So I just want to close by acknowledging that there's a lot of people who I'm working with and are I'm very grateful to for both the CHESS study and COVAX HIV. And then I'll thank you for your attention and happy to, with Curtis, answer any questions that might have arisen.
Thank you both, Curtis and Anne, for a great presentation of a primer of the immune system, humoral, cellular, and what studies we're doing. One question we have, I think for many people living HIV is in mind, as some people may qualify for a third booster shot, is it a good sign to mix various vaccines depending on what people got before, whether it's Pfizer, AstraZeneca, Moderna, like you're saying, well, one is a vector, one is mRNA, should I be mixing that? Will I get a good boost out of it? What are your thoughts on that?
Sure, it's an important question, and I think we're all aware that this has been one of those dominant stories in the news. I think, to be honest, I think we've spent too much time worrying about mixing and matching. Ultimately, they all generate good antibody responses, and they all appear to be safe. There's no evidence that receiving one followed by a different one is going to diminish your immune response. If you received the AstraZeneca vaccine, there is some data that you will get a very good boost in your immunity by getting one of the mRNA vaccines. But no evidence that there's any safety concerns with mixing and matching. Keep in mind, we've been mixing and matching different vaccine brands and types with other diseases. Influenza is an obvious one. Hepatitis B is another one. And we've been doing this and there's been no problem. So I think, I wouldn't go out of my way to say I have to get this vaccine because I got the same one before.
So no fighting over the choice of vaccine.
Yeah, I think there's other things that we can expend our energy fighting over.
I have a question from Kiro. So for some people who had bad side effects from the first 2 doses, should they consider getting a third dose? Would the benefit outweigh the negative outcomes?
Yeah, I mean, that's an important question. And with these vaccines, a lot of us have got reactions, and there's a whole spectrum of reactions. In my case, it felt like somebody hit me with a baseball bat in my arm for a couple of days. Other people, they feel pretty, flu-like for several days, and that's important. That affects people's quality of life. But the likelihood of very serious side effects and complications for these vaccines is really vanishingly low. And I can say that based on millions of people worth of doses of these vaccines. And you have to weigh that against the risks of getting COVID. The vaccines protect us from severe disease, but you can still become infected. That can result in you exposing other people. We don't want to do that, so that's a good reason to get vaccinated. There's still a risk for getting ill with COVID, even if you're vaccinated, so a booster could help. So I would support getting a booster, even if you had some of these local or systemic symptoms. Now, if somebody, in a specific case said, look, this knocked me out for a week and I had to go to the emergency, and there may be specific cases where I might look at the pros and cons and say, okay, in your case, maybe it makes sense to not get another booster or delay it to a maximum amount. But I'd say by and large, I think the benefits continue to really outweigh the negatives of getting booster vaccines.
Thanks, Curtis. Another question is, as people are planning to return back to quote unquote normal in March 22nd next year, and for past folks who are going back to work and going back to work offices, are there any specific takeaway messages like, is it okay to go back? Do you still have to maintain social distancing, maintain mask wearing, travel plans, people planning to travel, everyone's getting tired of being stuck for 2 years in the country, or the province. Anything specific, or could they relax personal rules if they got 2 or 3 shots?
I'll say a couple of words, and then I'm sure Anne has some thoughts about this as well. I think the best approach is a slow and steady approach to trying to return back to the way we did things pre-COVID. You loosen up the restrictions a little bit. You monitor the caseloads. If you see that they're spiking up, then you reinforce your restrictions and your public health practices, and you kind of titrate things slow and safe. You don't sort of declare the pandemic over and open up everything wide open and then don't bother to continue to watch what the consequences were. If you do that, then you end up with some of the situations we've seen in Western Canada and down in the States. So, slow and steady wins the race. I think we should slowly see, as more people get immune, then we can say, okay, well, let's increase the numbers a little bit, and maybe you don't need masks in this situation, but let's not charge into it.
And I'm sure, so go ahead, Anne.
I was just going to say I agree completely with what Curtis is saying, that I think following the guidelines around social distancing, masking, vaccination, I think that people should feel okay going out and doing things within those guidelines, but to continue to monitor them.
One final question is that yesterday, I'm sure everyone saw the news that the UK Medicine and Health Product Regulatory approved an antiviral molnupiravir for people who test positive for COVID and taken within 5 days of onset of symptoms. So once Canada approves it, anything specific for the HIV population to think about, like in terms, I'm sure there's not any drug interaction studies done with antiretroviral therapies, if you can take it if you're on. Is there something to think about?
Yeah, I think, thankfully we have lots of different strategies to protect ourselves. We have masks and public health practices. We have vaccine, and it looks like we are soon going to have well-tolerated, easy to administer oral medications that we can take at the time that we're diagnosed, that will protect us from being hospitalized and getting really sick. So there's a Merck drug that I think that's the one you're referring to, and
yep.
Pfizer has just put out a press release with a ritonavir boosted protease inhibitor. I know none of us really want to see the return of ritonavir, but this medication does seem to protect people from being hospitalized. So there will be these meds, which will be useful for protecting against big community-wide explosions and outbreaks of new COVID, and will hopefully keep people healthy and out of hospital. So it's just another tool that we'll have to protect ourselves.
Wonderful. Well, we're at the 12 o'clock mark, so I want to take this opportunity to thank both of you for your brilliance and your knowledge and your research work that you're doing for the community and for the people infected and affected by HIV. So thank you so much, and a big applause and much appreciation from all of us here. And keep us posted on your research findings.
Pleasure, and it really is an honor to be asked to to participate in your meeting, and good luck with the rest of the day.